Hi, welcome to Nagesh Biology online classes. Those who have not subscribed to my channel, please subscribe, like and share to your friends. Now I am going to give the detailed information about ETS. Electron transport system, it consists of 5 complexes. Among all these 5 complexes, 4 complexes play important role in transport of electrons, but whereas complex 5 that helps to synthesis of ATP, that is due to consisting of ATP synthase enzyme. How exactly this ETS mechanism works in eukaryotic cells? These total 5 complexes embedded in inner mitochondrial membrane. The mitochondria, it consists of 2 membranes, outer and inner membrane. But all these 5 complexes, they are embedded in the inner membrane of mitochondria. This first complex, it is complex 1. Complex 1, it has 2 prosthetic groups, that is FMN, flavin mononucleotide, and iron sulfur protein. This FMN located towards matrix, iron sulfur protein located towards intermembrane space of the mitochondrion. This FMN iron sulfur proteins, these 2 components are complex 1 components. So this FMN, what it will do? It is otherwise known as NADH dehydrogenase. So because of oxidation of NADH H plus, that is oxidized to NAD plus. Once carefully observe, FMN that is located towards matrix, NADH plus H plus that is formed in the during link reaction and Krebs cycle, that will come to nearer to complex 1, because complex 1, it helps to oxidation of NADH H plus. Because of oxidation of this one, it is oxidized to NAD plus. Once carefully observe here, because of oxidation of this NADH, what happens? 2 electrons are released. That electrons are accepted by FMN, that is located towards matrix. First electron acceptor, whatever electrons released from NADH H plus during oxidation, that electrons accepted by FMN, that is flavin mononucleotide. That electrons that is transferred to or transported to iron sulfur protein, that is also component of complex 1. Carefully you observe here, along with 2 electrons, 2 protons, that is translocated from matrix to intermembrane space. Now here, because of oxidation of NADH H plus, what happens? 2 electrons are released, 2 protons are released. These electrons are accepted by FMN. These electrons transfer to iron sulfur protein. So whenever electron transfers from FMN to iron sulfur protein, because of liberation of energy, whenever electrons move from one electron carrier to another electron carrier, energy is liberated. That energy helps to transport of 2 protons from matrix to intermembrane space. Intermembrane space nothing but the space which is present outer membrane and inner membrane, between space is called intermembrane space. But most important thing in our NCERT book, so there are 4 protons are there. You will get one doubt. At the time of movement of electrons, so here through like this, so there will be one more proton carrier will be there, hydrogen carrier will be there. That hydrogen carrier that is located in the complex 1 through this electron carrier, two more protons transports into perimitochondrial space. That is possible in only complex 1. So finally here, how many protons enters into the perimitochondrial space or intermembrane space? Through complex 1, 4 protons. But there is chance to confuse. So for understanding purpose here, I am explaining separately like this. But in the NCERT book directly they give 4 protons. There is chance to confuse. Sir, here while passing 2 electrons, how it is possible to translocate outside 4 protons? There will be inside the complex 1, there will be one more hydrogen carrier will be there. That hydrogen carrier, 2 more protons from matrix to intermembrane space, it transports directly. In our NCERT book you can see direct 4 protons. So that is very important. Next, now at present electrons are accepted by iron sulfur protein. Now at present electrons are accepted by iron sulfur protein. Now these electrons enters are taken by ubiquinone. Now electrons are accepted by ubiquinone. This ubiquinone, it will reduce to UQH2. Again one more doubt, from where protons comes to ubiquinone? So protons comes from 2 more protons accepted by ubiquinone from matrix, because it is mobile electron carrier. So this ubiquinone takes electron, 2 electrons are coming, no? So these electrons, it takes, ubiquinone takes 2 electrons from iron sulfur protein and 2 protons from matrix of the mitochondrion. Then 2 protons, 2 electrons accepted by ubiquinone. Then it is reduced to UQH2. Reduced ubiquinone is called ubiquinol. Now ubiquinol is formed here. This ubiquinol, here carefully observe, this is complex 3. Ubiquinol transfer electrons to complex 3, not for complex 2, because complex 2 function is different. So that will accept electrons from FADH2. So here this, so complex 3, it consists of 3 components: cytochrome B. Next, it also consists of movement of electrons, that is important, iron sulfur protein. Next, cytochrome C1. These are the components which are found in the complex 3. Ubiquinol, this will transfer electrons to cytochrome B. I am talking about electrons. Ubiquinol, that is reduced ubiquinol, that will transfer electrons to cytochrome B. That is part of the complex 3. These electrons, the 2 electrons, but one by one. So now I am giving the clarity about here. 2 electrons are moving so continuously, but here we took only 1 electron, but one by one like this, because 1 electron accepted by that component, so it will reduce, then immediately oxidize. Reduction oxidation continuously occurs. So that's why here we are taking here 1 electron. Now these electrons, whatever coming from here, from complex 1, these electrons transfer to cytochrome B. From there 2 electrons transfer to iron sulfur protein and cytochrome C1. Carefully you see here, whenever electrons are transferred to cytochrome B, here how many protons are released? 2 protons, no. These protons, so that will transfer to intermembrane space. I am talking about these 2 protons. Next, at the same time here, at the time of this ETS mechanism, now here oxidation of Fa DH2 also takes place near complex 2. Because of oxidation of this one, what happens? FAD plus is formed. This is complex 2, succinate dehydrogenase is there, no? Because of this succinate dehydrogenase, oxidation of FADH2 takes place. So here this one it consists FAD and as well as iron sulfur protein. FAD is there and iron sulfur protein. These are the components of complex 2. But carefully observe, because of this what happens here? Electrons are released. Electrons are accepted by FAD, flavin adenine dinucleotide. But most important thing, complex 2, through complex 2 there is no chance to movement of protons into intermembrane space. So that only it can accept electrons from FADH2 during oxidation process. So that electrons are accepted by FAD. These electrons are absorbed or taken by iron sulfur protein. That here ubiquinone, it is mobile electron carrier that helps to transfer of electrons from complex 1 to complex 3 and as well as complex 2 to complex 3. That is the specialty of ubiquinone. It is mobile electron carrier. These electrons, whatever coming from iron sulfur protein, that electrons accepted by ubiquinone, then it will converts into UQH2, ubiquinol. Again one more doubt, 2 more protons from where it comes? From matrix. So 2 protons takes from matrix, electrons it takes from iron sulfur protein, that reduced to UQH2, that is ubiquinol. So same whatever process here, same mechanism occurs here also. Now this will transfer electrons to cytochrome B. So this one ubiquinol is then it will transfer electrons to cytochrome B. Most important thing here, again 2 more protons are released, no? That protons, again here 2 more protons, that's why here these two protons coming from here and 2 protons coming from here. So these protons, 4 protons translocates into perimitochondrial space or intermembrane space. But here because of this one electrons transferred to cytochrome B, protons, 2 protons, here 2 more protons coming from ubiquinol. So here 2 protons from here, 2 protons from here. Total how many protons? 4 protons. These 4 protons enters into intermembrane space. So now most important thing here, these electrons at present here, electrons are moving like this. So it is complex 3. So this cytochrome B takes electrons from complex 1 and as well as cytochrome B also takes electrons from complex 2. These electrons transfer to cytochrome C. So reduction oxidation continuously occurs. Once it undergoes reduction, then immediately it oxidizes by donating electron to neighboring compound. So here these electrons accepted by cytochrome C. Most important thing, cytochrome C, mobile electron carrier that is located between complex 3 and complex 4. It is complex 4. So complex 3, complex 4, between mobile electron carrier will be there, cytochrome C. This cytochrome C peripheral protein, it is present on the inner membrane towards peripheral, towards intermembrane space. So this is total peripheral protein that is helps to transport of electrons from cytochrome C1 to complex 4. In the complex 4 there are few components are there. It is complex 4, no? First copper center A. Next, cytochrome A. Then cytochrome A3. Then copper center B. So electrons coming from here, cytochrome C1, these electrons moving like this. Now first electron accepted by copper center A from cytochrome C. This cytochrome C mobile electron carrier, no? Now whatever electrons coming like this, that is accepted by copper center A. Initially this copper center A accept electrons from cytochrome C. From here to electrons that will enter into the cytochrome A, then cytochrome A3, finally electrons enters into the copper center B. Two copper centers are there, copper center A, copper center B. So finally from this copper center B, electrons, now we will take two electrons, because so first two electrons coming from here, complex 1, next complex 2 also two more electrons are coming, but two electrons, so 2 electrons plus 2 protons plus half O2, all these oxygen final electron acceptor and final proton acceptor in the ETS. So electrons are coming like this, no? So 2 electrons, 2 protons, half O2 molecule, all these leads to formation of water molecule. During respiration, especially in ETS, only water molecules are formed. But here for 2 electrons, one water molecule will form. But here 4 electrons comes, no? From here 2 electrons, from here 2 electrons, 2 water molecules are formed. Here number of water molecules, so equal to the number of NADH and FADH2 which are oxidized in the ETS. One NADH if it oxidizes, one water molecule will form. One FADH if it is oxidizes near complex 2, one more water molecule is formed. Final ultimate electron acceptor and as well as proton acceptor is oxygen. So here water molecule is formed near complex 4. This total, already here this complex 4 also, it should translocates outside 4 protons, but already 2 protons are used, no? So remaining 2 protons, so this helps to transport of 2 more protons here. So 2 protons are used here, only 2 protons translocates outside, that is intermembrane space. Complex 1 and complex 3, complex 4 only helps to translocation or transport of protons from matrix to intermembrane space. Continuously what happens here? All these protons accumulate in the intermembrane space. Number of protons increases in the intermembrane space. Finally what happens? Here it is complex 5 is there. Complex 5 it has F0, F1 particle. This one it consists one enzyme, ATP synthase enzyme will be there. ATP synthase. This ATP synthase enzyme helps to synthesis of ATP. But here whenever 2 protons like this passes through F0, F1 particle, it is just a channel. So here it acts like a proton channel, F0, F1 particles. So this one complex 5 ATP synthase, here whenever 2 protons transports through F0, F1 particle, so energy is liberated. That energy helps to joining or combining of ADP and inorganic phosphate, leads to formation of ATP. Now F0 and F1, F0 it is embedded in the inner mitochondrial membrane, but whereas F1 ATP synthase, that particle that is embedded in the matrix of the mitochondria. But here 4 plus 2, 10 protons are there. 10 protons if the passes, it leads to formation of 5 ATPs, 3 ATPs related to 3 ATPs are formed because of oxidation of NADH. 2 ATPs are formed because of oxidation of FADH2, because here, because of hydrogen acceptor, extra hydrogen acceptor which is present in the complex 1, because of that here 1 NADH H plus, so it gives 3 ATPs. Here 2 protons extra transported, no, that's why here 3 ATPs, but FADH2 it gives 2 ATPs. So number of ATPs also vary because of oxidation of NADH and FADH2. So what type of questions you can expect in the exam? FADH2, that is oxidized near the following complex, complex 2. NADH H plus, it is oxidized near the following complex, complex 1. FMN located in the following complex, complex 1. FAD located in the following complex, complex 2. 2 copper centers located in the complex, the following complex, complex 4. F0 F1 particle found in the following complex, complex 5. Sometimes they will give movement of the electron also like this. So here mobile electron carriers, only 2 only there, that is cytochrome C and as well as ubiquinone. These are mobile electron carriers. So here mobile electron carrier, one is embedded in the inner mitochondrial membrane. So here inner mitochondrial membrane, that is ubiquinone, but whereas cytochrome C, peripheral mobile electron carrier, that is present so above the inner mitochondrial membrane towards intermembrane space, that helps to transport of electrons from cytochrome C1 2 and copper center A. This is total information related to ETS, electron transport system.